that cytokinesis, or that cartoon I just showed you, shows how cellular division is done within the animal kingdom. In other words, if we looked at one of our cells under a microscope that was dividing, it would look pretty much like that. You'd see nuclear division first, and then followed by cytokinesis. Now, let me run something back there real quickly. When you get to the cytokinesis step, notice that the membrane is pinching in, like right here and right there. That pinching in part, as the cell is in the middle of cytokinesis, is called the cleavage furrow. And here are some photographs of some cells from an animal dividing. You can see the cleavage furrow starting right there. It almost looks like a piece of bubble gum that's getting stretched. The cleavage furrow gets deeper and deeper. In other words, you get more and more stretching until finally the cell membrane snaps and you get two separate daughter cells. There's a nice photograph of a dividing animal cell. There's the cleavage furrow right there. That's the nucleus in one daughter cell, and that's a nucleus in the other daughter cell. And that might be a mitochondria right there. It looks kind of like a mitochondria. In any event, as far as plant cells are concerned, it is the same basic pattern, nuclear division followed by cytokinesis, but their cytokinesis doesn't have a cleavage furrow. Can anybody hazard to guess why not? What? I think I heard it. Cell wall. That's the problem, because animal cells don't have a cell wall, so they can't pinch in their membrane to do make a cleavage furrow. But plants don't have that option because they've got that cell wall. And so, how do plant cells do cytokinesis or do division? Well, it starts off with nuclear division. That part's exactly the same whether it's a plant cell or an animal cell. But for cytokinesis, what the plant does is essentially just build a new cell wall in between the two daughter cells, like that. And that new cell wall that divides the mother cell into two daughter cells is called the cell plate. So plant cells do not have a cleavage furrow. They have a cell plate that actually divides the two daughter cells. But other than that, it's fundamentally the same process. It's nuclear division and cytokinesis. There's a nice picture of a plant cell with the cell wall there in green. Cell plate. Okay. Now, focusing in on the nuclear division for a moment, where the nucleus splits and moves to opposite poles, there are two types of nuclear division. One's called mitosis nuclear division, and the other's called meiosis nuclear division. And here's the essential distinction. Mitosis is a type of nuclear division where the daughter cells are going to end up with the same number of chromosomes that the mother cell has. And when we're talking about mitosis, almost always what we're talking about is a diploid mother cell that divides to make diploid daughter cells, like you see there. Whereas in meiosis, it's where nuclear division is where the diploid mother cell divides, the nucleus divides in such a way as to make two haploid daughter cells. And maybe I could make human cells as an example here. If this is mitosis, remember, human cells have 46 chromosomes, so two sets of 23. And each daughter cell then ends up with 46 chromosomes, two sets of 23. But in meiosis, each daughter cell only gets one set of chromosomes. So this daughter cell only has one chromosome number 1, and one chromosome number 2, and one chromosome number 3, all the way, so one set of 23, and the same with the other daughter cell there. Meiosis is sometimes called reduction division, and you can see why. You're going from 46 chromosomes, you're reducing the number to just 23 chromosomes, just one set of chromosomes per cell. Let's see now. Of these two types of division, mitosis is by far the most common in our bodies. The only spot you get meiosis is in your gonads. So in your testes if you're a male, where you're making your sperm, or your ovaries if you're a female, where you're making your eggs, is the only spot in your body where you're supposed to have haploid cells, and so that's the only spot where meiosis occurs. But all other division in your body is going to be mitosis. And maybe we can use this part of the diagram. So here's where we all started off life as a diploid cell. So it was mitosis, 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 mitosis that allowed that cell to grow into 4 trillion cells when we were a newborn. So every cell in your body is 2N because it's made by mitosis. And all further growth later on in your body is mitosis as you become an adult. And it's not just growth and development, it's also repair. And got a little scar here, a little cut there. And so the cells that form that scar tissue did mitosis growth to fill in the cut there. Or if you break a bone, the bone cells do mitosis to make more bone tissue to fill in the broken bone, to repair the broken bone. So it's all mitosis except for making sperm and eggs is what it comes down to.
There are genes that control mitosis.
Yeah, I mean, both of these processes are run by hundreds of genes, and we're going to talk about some of those genes here in a little bit. Okay, for the rest of this lecture, well, it's getting near quiz time, but for the rest of this lecture, and as we continue with this mitosis lecture next week, we're going to be just focusing on mitosis. Everything that's going to follow now is just going to be about mitosis. We are going to cover meiosis. There's a lecture on that I'm going to give, but it's going to be a pre-lab lecture in a couple of weeks. But don't put away your stuff yet. I want about 2 more minutes, and then we'll get ready for the quiz. Okay, now, so this is a chromosome, and imagine a cell is going to go through mitosis. It's a mother cell, and it's interested in dividing to make daughter cells. It's going to soon divide and make daughter cells. Well, before a cell can undergo mitosis, before it can divide, it has to do what's called replicate its DNA. And at a visual level, in other words, if you were looking at the chromosomes, here's what it looks like when a cell has replicated its DNA. Each chromosome goes from looking like this to looking like that. And so notice that what's happened to each chromosome. The chromosome went from having one chromatid, one big piece of double-stranded DNA, to having 2 chromatids, 2 pieces of double-stranded. They're both still attached to the same original centromere. That hasn't changed. But you've gone from 1 chromatid to 2 chromatids. And the new chromatid, this one right down here, is an exact photocopy of the original chromatid it had. Nothing's changed. No alleles have been no new alleles have been generated. If it had the brown color allele right there, you'll have the brown color allele right there. For eye color, if you have a blood type allele for blood type A negative there, you'll have the same allele for A negative right there. So it's an exact photocopy of the original chromatid. And you refer to these as sister chromatids, the 2 identical chromatids on a duplicated chromosome. And notice that the wording here is a little tricky. You say that the DNA has replicated, but the chromosomes are duplicated chromosomes. And any chromosome you see that has this X shape to it is considered a duplicated chromosome. And one final point here. A duplicated chromosome is still considered one chromosome, which at first is a little counterintuitive because you say, wait, there are two chromatids there. Yeah, there are two chromatids, but there's only one centromere, and one centromere equals one chromosome. If you're asked to count up chromosomes, count the centromeres. Don't pay attention to how many chromatids are attached. One centromere equals one chromosome by definition. So if I were to show you this and ask you how many chromosomes were here in this picture, count this as one chromosome even though it has two chromatids attached to it, and count this as one chromosome, and count that as one chromosome. Everything, well, just comes down to how many centromeres it has. Okay, that's a good stopping point. When we come back on Monday, we're going to talk about at a molecular level in terms of the enzymes involved, how cells replicate their DNA. Alrighty, so put all your